A marsupial built a wolf skull, not similar to a wolf skull, the same skull. December 1933, Hobart, Tasmania. David Flea, Australian naturalist and filmmaker, pressed his eye to a 16mm camera inside Beaumaris Zoo. The concrete smelled of damp stone and eucalyptus. The animal in the enclosure paced, turned, paced again. Flea kept rolling. The footage runs 80 seconds. In it, an animal moves with a gait that is almost right, almost canine. The striped back, the stiff tail, the narrow snout, everything points toward wolf. But the hinge of the jaw opens past 90 degrees. No dog does that. No wolf does that. Nothing in the order Carnivora does that. That animal was Thylacinus cynocephalus, a marsupial. Its closest living relatives are the Tasmanian devil and the numbat. Its nearest common ancestor with any wolf lived 160 million years ago. Before flowers existed. Before the continents finished splitting. And yet, Stephen Rowe, University of New South Wales, published a morphometric analysis in the Journal of Morphology in 2007. His team measured both skulls with the precision of forensic engineers, Thylacine and grey wolf. The findings stopped the lab cold. The two skull shapes overlapped more closely than either species did with its own nearest living relatives. The 7th of September, 1936, the last known thylacine died alone in its enclosure at Hobart Zoo. No keeper stayed. The temperature dropped overnight. Tasmania had granted the species protected status 59 days earlier. Robert Paddle, Australian Catholic University, documented that moment in his 2000 historical analysis. The last Tasmanian tiger. The records were thin. The death went largely unwitnessed. What Flea's footage cannot answer is how. How does evolution build the same structure twice, from completely different raw material, across 160 million years of silence between two lineages? A 2018 Genome paper has an answer. Not the answer you expect. The evidence is in the regulatory architecture of the Thylacine's DNA, and it changes what we thought we knew about how bodies are built. Convergent evolution. Every biology student learns the word. Dolphin and ichthyosaur, bat and bird. Two animals, no shared ancestry, arriving at the same shape through independent pressure. Tidy. Satisfying. Case closed. Except it isn't. Stephen Rowe at the University of New South Wales ran the numbers in 2007. His team measured carnivore skulls across dozens of species, length, width, zygomatic arch breadth, orbital geometry. They fed everything into a morphometric analysis published in the Journal of Morphology. The result was not what anyone expected. The thylacine and the grey wolf did not merely resemble each other. Their skull shapes overlapped more closely than either species did with its own nearest living relative. Read that again. The thylacine was morphometrically closer to a grey wolf than to a Tasmanian devil. Closer to a placental predator it had never shared a continent with than to the marsupial sleeping 2 drawers away in the same specimen cabinet. That is not convergence behaving normally. That is something else. Standard convergence models allow for similar shapes under similar ecological pressure. They do not predict this level of precision. The Tasmanian devil, the Thylacine's closest living relative, has a skull that is wide, blunt, built for crushing bone. The Thylacine's skull is long, narrow, built for a sustained chase bite. 160 million years of separation, and the wolf got there first, or simultaneously, or through some mechanism the model doesn't account for. The tidy explanation starts to crack. Rowe's own words, despite 160 million years of independent evolution, the Thylacine and the grey wolf show striking similarities in skull morphology. Striking, not approximate, not suggestive, striking. Two researchers can hold calipers against both skulls and produce measurements that fall within each other's margin of error. That is a measurement problem, or a biology problem. Nobody was certain which. Convergent evolution explains the shape. It does not explain the precision. So if the anatomy alone cannot account for this, what is happening at the molecular level? Here is what the 2018 paper actually found. Charles Fegan at the University of Melbourne sequenced the complete Thylacine genome and set it beside the grey wolf's. Not to confirm the resemblance, everyone already knew about the resemblance. To find out how, molecularly, it happened. The answer was not in the genes themselves. The thylacine and the wolf do not share the DNA sequences that build their shared skull shape. What they share are the regulatory elements, the non-coding stretches of genome that function as switches, controlling when genes activate, in which tissue, at what intensity. Fegan's team found those switches had been flipped, independently, in the same direction, in two lineages that had not shared a common ancestor for 160 million years. Life wrote the same instructions set twice, different paper, same words. Stephen Rowe at the University of New South Wales had already quantified the physical result in 2007 in the Journal of Morphology. Thylacine and wolf skull shapes overlapped more closely than either did with their own nearest living relatives, measurably, within morphometric error. Rowe called it remarkably consistent constraint. Fegan called it convergent regulatory elements. Both phrases mean the same thing. The body plan was not an accident, but a solution. Life found it twice, using completely different molecular tools, arriving at the same answer to the same pressure. Remember the two skulls from the opening? Same orbit depth, same canine angle, same sagittal crest. The Genome paper explains why. Not similar pressures producing similar results, but the same molecular architecture reconstructed from scratch 160 million years later in a marsupial that had never been a wolf and shared no recent ancestor with one. Call it what you want. It means the solution space for a pursuit predator's skull may be so constrained, so narrow, that evolution has only one real answer, and it finds that answer regardless of where it starts. The footage Flea shot in December 1933 cannot tell you any of this. 80 seconds of a living animal, pacing, grainy, flickering, gone 3 years later. The genome is what remains, and it carries a question biology is still working out how to ask. If you found this fascinating, subscribe. We investigate strange, real creatures like this every single day.